Okay, let's get started then. I welcome all the audience, and I also welcome Markus Deserno, who is coming from the Department of Physics at the Carnegie Mellon University in Pittsburgh. And he started his scientific career in Europe at the Max Planck Institute for Polymer Physics, and moved over to the United States a couple of years ago, I think it was. And he is one of the pioneers of doing coarse-grained simulations in contrast to atomistic simulations. And this was really an area that I think the polymer physicists were the people that really started this. Now it's brought over into biological problems, and he will talk here about biological membranes. Welcome.
All right. Thanks, Olive, for the introduction, and thanks for the invitation. In fact, I have a little bit more work to do tomorrow, but right now I'm giving a talk here. It's a wonderful thing. I mean, I think every physicist in the world aspires once in their life to be invited to Stockholm, maybe for other reasons, but I'm here now, so it's a good thing. I should also warn you that since I came this morning with a red-eye flight from Pittsburgh, it might happen that I spontaneously fall asleep, in which case some of you has to come and wake me up again, and then I'll finish my talk. So what I'm going to talk about today is coarse-grained simulations of membrane phenomena on the kind of mesoscale, by which I mean 100 nanometers plus minus. And the talk is divided into sort of two parts. In the first part, I'm giving you my main philosophy, how I think about these kind of problems, why I think it's a useful way to think about these problems in the way that I do. And in the second part of the talk, I'm going to show you examples, in fact, on two different levels of resolution, how to describe mesoscopic membranes phenomena the way we do and what you can learn from this. In some sense, I think the first part is more important than the second part because I like ideas better than results. Which also means if for whatever reason I run out of time towards the end, there's no harm stopping. Which also means you can stop me at any time to ask questions, that's no problem. You might have to shout because I might not see you. So let's get started with what is it that we do. What we do is we look at things such as this. So this is a cut open version of a cell as you find it in any cell biology book that you might find on this planet. It's typically chapter 1, it's a cell. You see this is evidently organic. It has all these kind of wiggly, complicated, curvy things. And it tells you how complicated things are going to be. And if you actually keep reading the book, you realize that you're really suffering from an underestimation of how complicated things really are. Either way, so this is what you first encounter of a cell. And one thing you might remember, you might realize when you look at these things, is that almost everything you see in these things are membranes. And membranes is not just the outer envelope of this thing. If it's a eukaryotic cell, then there are lots of other organelles inside there which are membranes, such as the cell, nucleus, the endoplasmic reticulum, the Golgi apparatus, mitochondria, transport vesicles, and they're always with fancy names. All of these things are membrane organelles. And some of the reason why the stereotypical picture of a cell is a jar stocked full of membranes is the same reason that the stereotypical picture of a chemistry lab is a room that is stocked full of flasks and vials and bottles and beakers and test tubes, because in these little compartments, that's where a cell does its biochemistry. It's not all in one jar. It's subdivided in many complicated ways. And when a chemist does these things, test tube here, test tube there, let's put things in one, you can just do it like this, but a cell will have to do more complicated ways of getting stuff from A to B and fusing A things with B. So what you realize is that there's lots of topology changes going on in these compartments, which is sort of the cell's way of how to mix stuff when the cell wants to do biochemistry. And in order for something like this to work, a couple of things have to hold. One thing that has to hold for these constant reshaping, let me back up. So in order to work, the cell needs to constantly reshape its membranes. And that costs energy. It's a good thing that it costs energy because you don't want that to happen spontaneously because you want control over it. And the available energy is biochemical, and that typically means it's coming from ATP hydrolysis, which under physiological conditions gives you about 20 kT per hydrolysis of an ATP molecule. And in order for these processes to work, That means that the elasticity of the membrane that needs to be bent and deformed for these kinds of deformations and fusion and fission processes, the elasticity of the membrane somehow needs to match the available energy from that ATP scale. And what does that imply? Let me walk you through what that implies. So as far as energy scales is concerned, the one important energy scale in the membrane is its bending modulus. In fact, it has units of energy. It's basically the energy per unit area to create a certain rate of curvature in the membrane. Take this thing, you bend it, how much you have to pay for that? And then you might express this thing in terms of other known or semi-known quantities, looking up in books such as this. These quantities are the Young's modulus of the material that you're bending, and the thickness cubed of that material, and of course I'm already heavily simplifying, and so bear with me for a second. Let's assume that what I just told you, that this energy matching happens actually on that level, so that the bending modulus is of the order of 20 kT, which miraculously it actually really is. Then we can compare from the left-hand side to the right-hand side of this equation by making further use of the empirically known fact that the thickness of membranes is about 4 to 5 nanometers. It's a bimolecular layer of these lipid molecules. I can solve this equation for the Young modulus and figure out that the Young modulus has to be something like 10 to the 7 Pascal. If you don't have your typical scale of what 10 to the 7 Pascal is in your head, I'm telling you, 10 to the 7 Pascal is sort of the Young modulus of rubber. And that's not awfully surprising, because what is rubber? Rubber is a mixture of polymers, maybe hydrocarbon chains entangled in some funny way with nanometer length scales, which is sort of what a membrane is as well, hydrocarbon chain, nanometer length scales. So that's not surprising. It's soft matter. Membrane is made of soft matter. Let me do a little thought experiment. What if I say, okay, nowadays, 21st century, mankind can do all kinds of super cool nanotechnology. Let's build all of this with super cool nanotechnology, but let's make it with cool nanotechnology material. Let's do it, instead of with rubber, let's do it with steel. Steel has a different Young modulus. It's 4 orders of magnitude higher. It's about 10 to the 11 Pascal. And if I still insist on that kind of energy matching, that I want an energy substantially bigger, but not awfully bigger than the thermal energy scale for the bending rigidity of this material, that would tell me something like how thick it can be. And the answer is it would now have to be 2 angstroms thick. And that of course means, well, a material such as this would fall apart, to fail. I only know of one material that's basically atomistically thin and doesn't fall apart, which is graphene. Okay, nature doesn't make its membranes out of graphene. There's a deep reason why nature chooses soft matter. I mean, sort of the moral of that story is nature has to use soft matter if it wants stuff that is thermally stable, so this is bigger than kT, but not so stable that you can't deform it with available energies on that local scale. And that means that has further implications. The fact that membranes must be made of soft materials, which nature figured out 4 billion years ago, means that we will invariably encounter very long timescales if we study these things. I mean, long, let's say, from an atomistic point of view, if you want to do atomistic simulations. And let me walk you through this as well, by taking one example that you, at least one person in this room is very familiar with. That's a simulation done about 10 years ago by Eric Lindahl and Ole et al. About an atomistic simulation of a lipid bilayer, which was about 20 by 20 nanometers. It contained about 1,000 lipids, and it was pushed forward for about 10 nanoseconds. Now, I should say that was a really Herculean effort back then. And they looked at fluctuations of this thing, determined bending rigidities from the fluctuation modes, and so on. I'm going to talk a little bit about that later. But for the kind of stuff that I'm doing, looking at, let's say, proteins in membranes, and more than just one protein in a membrane, how do they interact, what do they do on a mesoscale of 100 nanometers, I might ask myself, okay, 20 by 20 nanometers is still a little bit too small for the kind of stuff I'm interested in. I would like to do, let's say, 100 by 100 nanometers, which is bigger, so I have to do more work. How much more work would I have to do? Okay, let's look at this. Let's generally ask the question, how does the computing effort for a membrane of size L scale with L? So that there's a simple and a complicated answer to this. The simple answer would say, well, evidently the area of the membrane scales with L squared. Since that tells me how many lipids I have to simulate, the effort should go up like L squared. But that's not true. And the answer for this, well, why that's not true, is that it's not enough to just stuff your membrane in your computer. Once it's in the computer, you also need to equilibrate it. Because what you're looking at the end of the day, okay, what I'm looking at most the ends of most of the days, is equilibrium properties, which means I need to sample this thing, fluctuating around, until I have a sufficiently good statistical average of what I'm looking at. And that means I have to wait until all these slow modes have decayed. So if I make this thing bigger, it has softer, slower modes, they take longer to decay, so I need to simulate it longer. Well, how much longer? Well, the answer is for these kinds of systems, in fact, for the kind of system that I'm looking at, where there's no solvent in it, I'll explain this in 10 minutes or 5 minutes, it turns out that the relaxation time of the longest node, which evidently scales like the center of the system, grows like the fourth power of my system size, like L to the power 4, and that's multiplied on top of that. And that means that the overall effort doesn't scale like L squared, it scales like L to the power of 6, and that suddenly looks intimidating, and it should. Because it means if I want to go from, let's say, 20 nanometers, as all I did 10 years ago, to 200 nanometers, as I'm excited about for whatever reasons right now, I have to do 1,000,000 times more work. And let's all remember, 1,000,000 is 10 to the 6, so that's 2 to the 20, roughly. And if Moore's law holds that 20 doublings of computer power with about 22 years per computer power doubling means 40 years. Since calculating from 2000, that would be 2040, but I'll be retired by then. Besides, it's really hard to find a PhD student when you tell them after 40 years you're going to have your result and they're going to write your thesis. They kind of don't like that. Well, not in the US. So I have to be smart about this. I have to figure out a way of how to look at these large scales without being beaten to death by increasingly strong equilibration problems. That's one of the reasons why I do what I'm doing, which is coarse-graining, which is simplifying the system and throwing away a lot of detail so that what's left I can spend more time on, and I can look bigger systems, I could run them longer. But that's only one of the reasons why I'm doing this. There's a second reason which people don't as frequently mention. The second reason is I want to gain insight. And that being, if there's stuff in my system I can throw away and still get a good physical answer, then whatever I threw away apparently wasn't that important to get that physical answer. And that's really, as a physicist, what I like to understand. What is it that I can throw away? And this insight question kind of reminds me always of this famous person. Does anyone recognize? Probably no one knows. This is Richard Hamming, one of the founding fathers of computational methods in sciences, who said the purpose of computation is insight and not numbers. And I think that's a really awesome statement, and I think it should underline everything that we do as computational scientists. You might say, well, that's true for physicists, it's not true for engineers, they really want the numbers. But the most famous book that he ever wrote was called Numerical Methods for Scientists and Engineers, so I think he thought about this for a while. By the way, you can also tell that's a long time ago because the jacket is a little bit outdated, I think. And these things here back then, these seem to be, that's before my time. So, we have all the justification and I can pull authority for what I'm doing. So what am I doing? I do membrane simulations, but I do it in very simple ways. Meaning, okay, in fact there's a whole range of varieties of how you do membrane simulations. You can do membrane simulations of this type, as we just seen before, the atomistic simulation where I lovingly take care of every single lipid, like something like this, and we get something 50 or so, depending on how I count, 50 atoms per lipid, and then I can slowly combine different chemical groups and moieties that are, let's say, the chemists give them a distinct name, they say, that's a choline group. Okay, you call this a choline group, and you look at this as an entity, or I can make a bead and call it a choline bead. And then I make my lipids out of these beads. And once, this is a slippery slope, right? Once you start that, you can, okay, I call this a head group, I call this a head group, I call this a head group. Well, there's just a head and a tail, I'll just paint a sphere blue and yellow on two different sides. Or I just say, well, at the end of the day, membranes make surfaces, or just look at triangulated surfaces. There's a whole spectrum of levels of description I can choose for membranes, from increasing detail to increasingly coarse-grained, much detail, little detail, and of course there's lots of people everywhere who've worked on these things. I'm going to be interested more in this intermediate regime down here, which can again be subdivided by all kinds of buzzwords, standard energy, DPD, water-free, which you don't have to remember at all. And again, names. What I'm going to do today is I'm going to focus on two levels of resolution that we have worked on. One is this level of resolution, which is basically 3 beads for a lipid, and one is this level of resolution, which is sort of a couple of beads, more than 3, but less than what you really need, per lipid. I'm going to start with this one, and if for whatever reason I run out of time and I have to skip this one, then so be it. It's not that important after all. And anyway, is anyone going to warn me when it's like 5 minutes before cutoff or 10 minutes before? Good, thank you. Because I don't see a clock here. But there's one. Good. Okay, let's start with this one. So meet our lipid. The fadey shaded thingy in the background here, that's like what a real lipid looks like if you download the picture from Avanti. The one thing that this illustration should show you is that what I'm taking account when I model my lipid like this is that I say, okay, there's a sort of region up here, which is the head group region of the lipid, which has the property, turns out, of being very well water soluble, while these regions down here are very different. They're basically hydrocarbon chain, they're oils, they're not water soluble at all, so they're different. So I'm going to account for this bit of the lipid different, and for this bit, which for the purpose right now means I color them differently. Really what it means, they're going to have different interactions. Notice also that roughly I'm getting the aspect ratio right of how wide a lipid is compared to how long it is. Anything else really is absent after I've done this kind of gross simplification. Since of course atomistically every one of these single atoms has some kind of funny interaction with every other one of these funny single atoms, and I know what they are in some sense because I know how carbon interacts with something else, that's fine, but I don't know how the blue bead interacts with the yellow bead. There's various ways of how I can come up with these potentials. What we did is a cheap way, we just picked them out of thin air with a physicist's eye, however, because we know what they're supposed to do. The idea is these lipids in water aggregate to make membranes. Why do they do that? Because the tails don't like to dissolve in water, they don't like to be surrounded by water, so they all come together and pack nicely to keep the water out of this kind of tail region, and then you get a membrane. Well, in our model, this is really everything that's in there. I'm not missing anything. There is no water in this particular kind of model. But if in the real world, the water drives the aggregation of my tails by this so-called hydrophobic effect, once I throw water away, why would these things ever come together? The answer is because out of thin air, I picked an attractive interaction between them to make sure they do come together. All right? So how do you choose this? Okay, that's a matter of choice, and people, in fact, have randomly picked from thin air for about 20 years potentials in order to do that. Actually, they all have always picked the same potential, have always picked a Lennard-Jones potential, because if you don't know what you're doing, you will always first pick a Lennard-Jones potential. And that didn't work. So there was 20 years of Lennard-Jones fudging going on. Between the heads, since they don't need to attract, they bounce into each other and repel, there's nothing else going on. It turns out, so if you do this kind of thing, you have basically a two-dimensional quote-unquote phase diagram that you can draft up as opposed concerning this kind of funny attractive interaction. And that phase diagram would say something like this. Okay, this interaction here has some sort of range, I don't know what it is, some range, and it has some sort of depth, okay, range and depth. Let me plot the range really as range and the depth as temperature divided by depth so that I have a temperature scale that looks like physical. If I do this and I try all the points in here, I realize if it's very hard and I don't have much of an attraction, the thing never aggregates. So I have some unstable lipids floating around. We could call it a gas phase or something like this. If the temperature is very low, lipids do aggregate and they form a fairly rigid so-called gel phase. And somewhere between the gel phase and the gas phase, there's a fluid two-dimensional phase of lipids, and that's what everyone's interested in, because these are the phases of membranes that matter in biology. It also turns out that this fluid phase doesn't exist everywhere. It exists if your attraction range is bigger than something, or 0.7 in God knows what units. But it turns out in these same God knows what units, a Lennard-Jones potential would be roughly here, which means if you tune your Lennard-Jones potential, you either get a gas phase or a rigid gel phase, but you never get the fluid. So these 20 years of picking out of thin air were simulations that just tried to find a fluid between these two phases and didn't kind of. The fluid's here. You need to make the interaction a bit longer range, which is not quite obvious to begin with. The deeper reason later on you can rationalize this by saying, okay, some of these long-range interactions somehow help to save the system some entropy, which you need in a fluid phase. And in retrospect, I mean, as I told you, 20 years of people, why is it working? There's always papers that you later realize you could have looked at, such as those. They would tell you have to increase the range and then things become fluid. We only realized this again after the fact. But anyway, there's some justification for why that works. In this case, it also tells you something. I mean, people who construct coarse-grained models such as this, just picking a potential from thin air, which is called empirical or top-down. Occasionally don't think very much it matters what kind of potential you pick. Oh, I need something that has a hardcore repulsion and some sort of attraction. I'm going to pick Leonard-Jones. In this case, it matters the shape of the coarse-grained potential can be qualitatively important in the sense if you pick the wrong one, the phase you're interested in might not even be there. So that's a little warning that top-down modeling might occasionally not give you the right stuff. But since we now understand what we need to do in order to get fluid phases, we kind of feel comfortable. So what I'm going to do now, I'm going to illustrate in a few ways why doing something like this will give you systems that, to a large extent, justifiably can be called membranes. And I'll show you examples of what we can do with these things. So let's start with the material properties. So, okay, first of all, they do self-assemble. They make two-dimensional sheets. They are fluid. So in that sense, they look like membranes. But are they membranes? Are they elastically membranes? Well, the material properties that are the most important one I first talked about is this bending rigidity. How stiff it is, and is this really bending rigidity, or is it something else? This I can figure out in various different ways. One way to do this, in fact, in simulations or in experiments, is always the same thing. You can either look at how that thing fluctuates, or you can actively deform it and see, okay, wow, what's the force in order to deform it? Okay? The fluctuation bit basically works like this. You say, okay, I have a membrane that's basically flat. The deviations from flat are going to expand, and some height function is a function of position, h is a function of x and y. Then a curvature elasticity basically means I have a curvature modulus times a curvature squared. The curvature is probably going to be something like the second derivative. Forget about this term, that's a tangent, doesn't matter. So my Hamiltonian is an integral of the membrane bending modulus times curvature squared. Since this is quadratic in the relevant variable, the height function, I know instantly, oh, I have a harmonic theory. I can solve everything. I can Fourier transform this thing, and then I figure out that the mean squared amplitude of any Fourier mode that this thing could have is proportional to kt, well, because it comes from a fluctuation, and it's inversely proportional to things such as wave vector to the power 4, bending rigidity, and so on. If we forget the tension term, then this thing goes away. In fact, so let's get away with this, then the mean squared amplitude is kt over bending modulus times system size squared, and it decays like the fourth power of wave vector. What the first thing you do is, does it decay like the fourth power of wave vector? Because if it doesn't, it's not a curvature elastic theory. Okay, it should decay like the fourth power of wave vector for a large wavelength, because this is continuum. And you check that. And if it does, you fit the prefactor, and since you know everything except kappa, you get the bending modulus. And we did this, and we get a number, and we can tune it depending on what interactions we choose. In fact, we can tune it to reasonable values. The problem only is that these curvatures are not really going to be very big. You can convince yourself, in fact, that these are really, really small curvatures, because I told you the bending modulus is something like 20 kt, and these 20 kt, the energy fighting against the 20 kt bending modulus is 1 kt thermal energy. It's not going to bend it very much. But if I later want to do simulations with these membranes that involve strong bending, for instance, I'm going to create a whole vesicle out of this, it's very likely that these bending, that the radii of curvature I'm going to encounter then are just going to be 2 orders of magnitude higher. Maybe all of this is entirely irrelevant to 2 orders of magnitude higher up the curvature. So we decided we need a second way to test that. And so we came up with a second way of measuring the bending modulus by doing something I could call this like a rigatoni simulation membrane. You basically take a membrane, you roll it up to a cylinder, you spin this cylinder in your box, and then by the usual trick in computer simulations, you link this up over periodic boundary conditions, so this looks like cut open, but it really connects to whatever is on the other side. The nice thing about a system such as this is that every lipid is in exactly the same environment. This is translationally and rotationally invariant. Every piece of the surface looks like every other piece of the surface. The next thing is, if I have a fixed number of lipids in my box and all of them are essentially in the bilayer, the area of that bilayer is some number and it's fixed. What happens now if I take that cylinder and I stretch it? Okay? Since the area has to be fixed, making it longer means, of course, I have to shrink the radius, otherwise the area couldn't stay fixed. But since shrinking the radius means putting more bending energy into the system, the energy has to go up. Make sense? And if the energy goes up, I can ask the question, how could the system increase its energy? Well, the answer is, while pulling it, apparently I pulled against a force, And this force times distance is not going to give me work. This means I did work, and that's the work that increased the energy. But that means there had to be a force. This thing has to be under an axial force, and the origin of that force has to be bending. Well, then you realize, okay, I can write this up. The energy is bending modulus, curvature squared, times the area of this thing. The derivative of this with respect to length at constant area is my force, and it's just 2 pi times the bending modulus over the radius. Well, that just means the force is easy to measure in a simulation, the radius is easy to measure in a simulation, but both of these things are tremendously hard, by the way, to measure in the experiment. People have tried and done. But either way, this and this I can measure easily, so I can extract kappa. Now I can ask 2 questions. Is this kappa the same as the one that I got here? And if not, how is it different? Well, it turns out, okay, plots and stuff like this, you get the same result in both cases. But the interesting thing is in the second case, when I actively impose a deformation, I can impose a really, really strong deformation. What I've plotted here, let's look at just the inside, is the bending modulus extracted from cylinders with a certain curvature, 1 over that curvature. And W is the thickness of the membrane. So at this point, the thickness of the membrane and the curvature radius I'm imposing are almost the same. And the value I'm getting for the bending modulus is the same as something that is like an order of magnitude weaker here. And it agrees with what I got on this side. That's very, very amazing. There's slight variation within the error bars. You can't see that here. But it means I can sort of bend the hell out of these membranes, and it's still beautiful quadratic curvature elasticity, which is not what we wanted to show here. We wanted the quartic effects. But okay, what this tells us is we can describe our systems up to ridiculously high bendings with the existing simple quadratic theory which is out there. It's very convenient if you later want to do any comparisons. Alright, so you can do all kinds of other things. Okay, get the same answer. We can tune within the range that is experimentally relevant. Fine. So we have now self-assembled fluid 2-dimensional bilayer sheets that have curvature elasticity and the right bending modulus. Okay, we're pretty close to having a real membrane here. I can measure other things. I can measure expansion moduli of these and so on. What's the energy to stretch it? Okay, if we stretch it too much, I can pour. If I compress it, it buckles. There's theories I can write down for how the system should behave. I can fit these theories to what I see. Is everything fine? Okay. From these theories, I can also measure what line tensions are. The line tensions are in line with what I'm expecting. All of this looks good, which is, by the way, scary because I didn't tune any of this. It just came out. I can even look at things such as thermal expansivity. What if I make the system warmer? Everything jiggles more, the area per lipid should go up. And it goes up at a certain rate. I can measure what that rate is, and it turns out to be 2 times 10 to minus 3 per Kelvin, which is basically the experimental value, which is even more eerie. I think that's a fluke, but it turns out to be right. So I'm collecting evidence that permit me to call these things membranes. So, let's say we agree that I can call these things membranes. What can I now do with these things? The first thing I'd like to show you is a study where we said, okay, we have all these lipids. I can now change a little bit about the shape of these lipids and do the following thing. I can say, what if a lipid has a head and has these tails, but there's no reason that the head exactly has the same, let's say, width as the tails. You like to think, you can think of a lipid as having some sort of degree of freedom, such as this big head, small tail, or small head, big tail. And in which case, they would pack in funny ways when they try to aggregate into, well, aggregates that shield the tails from the water. There's a simple theory out there which tells you, okay, there's a packing parameter which I can define, which is the volume of the lipid divided by its length and the area of the head group. That's a dimensionless parameter. And you can, with sort of basic simple geometry, convince yourself that if this value is less than a third, then all of these things basically pack together and make one little sphere. They don't make bilayers, they make a little sphere. That's called a micelle. If it's between a third and a half, they make kind of long tubes like sausages. And if it's bigger than a half, they do make the membranes that you want. So now you can see what doesn't work. You can do simulations, and indeed you get the entire scan from micelles, or the cylindrical micelles of our membranes. And I'm matching this up with, how do I match this up? I can't easily measure these things. It turns out, once you permit me one fitting parameter, I can take one of these two boundaries, either this boundary or that boundary, and can pin it on one half. And then the location of the other boundary is predicted. And it lies where it's supposed to lie. So I now know that this degree of freedom is something I can tune. and I can take it again serious in the sense of what predictions of continuity theory should tell me what's going on. So let's use this degree of freedom and play with it in a context that actually matters for membranes. So here's the context. So we look at mixtures, membranes that consist of, let's say, this lipid mixed with that lipid, which physiologically means, okay, we have a mixture of a lipid that has a kind of a small head and a lipid that has kind of a big head. And we put these things into these kind of spherical, closed bubbles, which are called vesicles. Let's think about this for a moment. Where would they go? Well, entropy would say, well, this is all mixed, they're all going to be everywhere. But evidently, you would imagine that putting a big-headed lipid on the outside is going to be easier than putting it on the inside, because somewhat the curvature on the outside, everyone being curved like this anyway, is more accommodating for that shape than doing the same thing on the inside, where this thing would like to curve this, but the curvature of the monolayer is exactly the other way around, and vice versa for these lipids. So energetically, there's a driving force for these guys to go on the outside. Entropically, there's a driving force for everyone to go everywhere evenly, which means there's going to be some balance, there's going to be some non-trivial distribution. In fact, a simple model, which means the energy is just the difference between the monolayer curvature and the curvature of the lipid squared. The simplest thing that anyone can ever cook up is a harmonic oscillator. So that's my harmonic oscillator for how lipids couple to the shape in which they live. And ideal gas. This simple model tells me that the log of the concentration of big-headed guys on the outside versus big-headed guys on the inside is going to be this expression, which is going to be, first of all, proportional to the curvature of my membrane. It's more strongly curved, more strong difference. Inversely proportional to temperature will make it hot, that thing goes away. And it's proportional to lipid curvature. And the proportionality factor is this funny coupling constant, which, since I just wrote this down empirically, I really don't know what that is. But the prediction that the log of that ratio is linear in curvature is something I can test. So I go ahead and plot it and test it. And it works out. So the log of that ratio is linear curvature. The two plots I'm showing here are for two differently strong asymmetric lipid curvatures. So why would that matter? Why would I care about these things? Because there are papers out there, starting in the bioliterature, who ask the question, you know what, these membranes with all these organelles and endoplasmic reticulum and Golgi and blah, and all these names, they all have very distinct lipid compositions. And still, they're just constantly exchanging lipids all over, because that's how the cell does this. How is it that membranes can maintain these different distribution functions, like these lipids are here, and these lipids are here, and they stay where they are? How can a cell maintain these differences in the presence of all these constant exchanges? And one hypothesis how that works was, well, maybe the different organelles differ in the spontaneous curvature of the shapes of these lipids, and they just go wherever they like it. In other words, the spontaneous curvature of the lipid is a contributing factor to its sorting. Okay? That's a physical reason for why things might go rather in this membrane as opposed to that membrane. And I'm very happy to read these things in biology papers, because it means biology start to develop physical intuition about how things might work. But of course, it's not enough to claim this. Now you have to figure out how the effect is strong enough for that to happen. So now we can measure this. Evidently, there's sorting. There's no question about it that sorting is going on. The question, of course, really is, for curvatures relevant in biology, for the sizes of region, what remains of this effect? And the answer is, for realistic curvatures, radius of curvature being 50 nanometers, that's about as strong as things are usually bent. We're down here in terms of K, and that's a really, really small sorting efficiency. It boils down to something like a concentration difference of the lipids between inside and outside of about 3%. And that doesn't sound very much like efficient sorting. You would say, well, all you need to do is you need to increase the asymmetry of the lipids, and these curves steepen up, and you move this thing up. But if you increase the asymmetry of the lipids, then what I've showed you previously, what's going to happen is that these things make micelles. They don't make membranes anymore. So you can't play with that degree of freedom. So the best you can do is the 3% effect. Well, all right, so that's not what we wanted to find when we did this paper. We wanted to write this grand paper about we have figured out that it really works. So we wrote the paper a bit depressed and said, okay, it doesn't work. Later that was confirmed by very beautiful experiments from the group of Tobias Baumgart, who just did experiments basically of almost this type and said, yes, it doesn't work. And that puts the ball back into the court of the biologist and says, all right, so how does it work? It doesn't work like this. There's more physics you can think about it. Like, if only what you have is lipid shape asymmetry, well, then apparently not. But there's lots of other fields. You could have mixture degrees of freedom. You could have sizes, maybe lipids alone, not, but maybe proteins sort in this way. And it seems proteins do sort in this way. Just lipids are a bit too small for this. But lots of cool ideas have come from this, which we thought was nice. Let me show you something else. Okay, that was lipid curvature effect. Since we're talking about mixtures anyway, let's talk about a different mixture effect. So this is something really simple, and physicists understand this like since decades. So let me just show you a biology-relevant version, if you like, of an Ising model. So here's the thing. Remember, this was the kind of funny phase diagram that we had. You can tune interaction range, you can tune temperature, get phases. Let's assume we want to do really a two-component membrane, and let's call it lipid A and lipid B because we don't like these long names. Lipid A and lipid B. How could I create two different lipids? Well, with two different lipids, I'm going to have three different interactions. I have A with A, B with B, and A with B. What do I pick? Well, here's one thing that you could do. You could pick the homotypic interaction, which is A with A and B with B. You could pick them identical, but the heterotypic interactions, A with B, you pick them a little bit weaker in the sense the interaction range is not quite as long as for the homotypic interactions. What that basically means is that if you have two unlike lipids coming together, they gain a little bit less in energy than if two like lipids come together. And therefore there's a tendency to avoid that. And the best way to avoiding that is to just get all the A's on this side of the room and all the B's on that side of the room. That, however, is entropically not quite favorable because you have ordered your whole system now. So there's going to be a balance. The usual lattice gas argument, if you like. What you find is that if you plot this asymmetry, let's say the heterotypic interaction on this scale, you're going to have, for whatever reason, 1.6 is the ideal mixture. Then you couldn't even tell A and B apart by any means. If you now reduce the interaction range between A and B and make them more unlike, then you reduce it, then you get a situation where the system phase separates, and since it phase separates, it has a line tension that you can measure. So there's a line tension between, let's say, the blue phase and the yellow phase, and it has some value. If you make it now less asymmetric, go up again back to 1.6, the line tension drops and it vanishes at some point. And it doesn't vanish here, it vanishes here. So there's a whole region in the membrane where the system is mixed, but non-ideally so. And non-ideally so means if within that range you measure something else, such as a correlation length, okay, what's the distance that away from an A lipid that I remember there was probably an A lipid at the center, not a B lipid at the center. I call that a correlation length over which the system remembers its composition, and I plot the inverse of that, and this is a number that again vanishes here. In theory land, these meet at the same point. In simulation land, they don't, because it's very, very hard to pinpoint these things here. But bear with me that within the resolution that I can do, there's a point here which, for symmetry reasons, is a critical point in the system where this system phase separates. Why would that matter? Other than, I have found an Ising model, because this is not what it is, it's an Ising model. Why would that matter? It matters from an experimental point of view because there's very substantial evidence that that's what's going on in the cell membrane of a cell. It's known that that's a mixture. And for various reasons, people have argued that the composition of that mixture is very, very close to a critical point. In the sense that if I, for instance, lower the temperature a little bit, the system kind of spinodally decomposes. Which also means the correlation length at body temperature for these cells is maybe, let's say, 50 nanometers. It's not zero, or it's not like angstrom, like atomic length. It's 50 nanometers. It's suboptical, but it's large compared to the size of these lipids, which is nanometer. Why would that matter? Because here's a nice effect you can have in this way, and that's the following. Let's assume we add a third component to our system, which we call protein. And the protein sits, let's say, on top of the membrane. There's lots of examples in biology where proteins would like to sit on top of membranes. They like to be localized there. You can do nice biochemistry once you're two-dimensionally localized. You can transmit signals across membranes. Lots of reasons. Okay, let's say there's a protein sitting on the membrane, and for whatever reason, it rather binds to the A lipids and not to the B lipids. One reason you might think, for instance, why that might happen is that, let's say, the blue lipids, say, are negatively charged, but the yellow lipids are not. And the protein happens to be positively charged, in which case it would bind to the blue ones and not to the yellow ones. Now, in a situation where the lipid mixture is entirely ideal, you get these proteins stick to the membrane, and they sit here, sit here, sit here, sit here. Everywhere where they sit, there's a little blue patch because they bind to the blue and not to the yellow. But other than that, they're randomly distributed. But what if I get the membrane now into the non-ideal regime where they're still mixed, but non-ideally so? That means that every protein not only sits on the blue patch, but has a certain halo of blueness around it, which is an extension of the correlation length. And now if you get several of these proteins close to each other, these correlation lengths overlap. And the overall free energy of the system now depends on what happens with this overlap free energy. You can do not too complicated calculation to convince yourself, it's not trivial, but it's also not too complicated, to convince yourself that when you bring these things together, the overall free energy goes down. Halo overlap will make them attract. Well, then that's what they do. They come together and attract. Why does that matter? Because these inhomogeneities that people talk about, it might be these 50 nanometer correlation lengths, in biocontext have spurred, I don't know, 20 years and thousands of papers under the buzzword lipid rafts, which says the lipid is locally inhomogeneous. That might cluster proteins together, but people very often have thought about this as just ordinary phase separation. Okay, there's a little blob of A, which doesn't make any sense because once you get phase separation, the whole membrane should phase separate. It doesn't. But you don't have to make the whole membrane phase separate. If you are in this non-ideally but mixed regime, halos are sufficient to make proteins come together, which is really all what people who think about rafts care about, getting proteins together in domains. That's what can happen. So you can now do Ising model calculations for this. You can do the ground state, which we've done. You can do the Casimir corrections to these interactions, which was done, I believe, by Eisenriegler. You get enormously long-range interactions that just go beyond anything. The Casimir interaction here decays like 1 over the distance to the power of 1/4, which I don't know anything that's longer range than that. Which is cool, because it means these things talk to each other over length scales that are longer than any other physical interaction in the system. So these are the things that might bring stuff together. I think that's pretty cool. Good, so we've also went ahead and measured these interaction potentials, because you can measure pair correlation functions and all kinds of other things you want, and we didn't really do a spectacularly good job there, but we could see that these lengths are macroscopically large compared to anything else in the system. In fact, if we go pretty close to the critical point with this one, the interaction is so long range that even in a fairly big box of something like 100 nanometers, we couldn't get our proteins far enough apart to make sure the force goes to 0. So we couldn't even see this thing saturate. That's how strong the force was. Okay, now it's lipid mixture. Final example is protein-induced budding. Now we also have a mixture, but it's only lipids and proteins. And the story is basically this. I started off with explaining that you have these cells, and there's all these kind of funny interactions going on. Like bubbles and fusion and fission, and all of this has to happen. How does that happen? It costs energy, yes, we said when we adjusted the stiffness of our membrane to make sure that we can pay for that energy, but still it has to happen. Someone has to do it. Just because you have the energy doesn't mean something's going to happen. Okay? Just because you have enough kilojoules to do work doesn't mean you have the motor to do the work. So what's the motor in this case? Well, here's one speculation for what such a motor might be. Okay, everyone agrees that probably somehow proteins are involved. So proteins go there, they bend the membrane, someone brings all these proteins together, so make a bubble here, and they just go and make a bubble. But someone said, in fact, Anthony said a couple of years back in Current Opinions in Cell Biology, as a bio paper, and he's a biologist, that I can think of a mechanism of how they do that, where I don't need to have some other protein police, which is you go there and you make a bubble. I could just say, let's say there's a single protein sitting here, and it makes a little bubble, or a full bubble, just a little dent, and another protein here, which makes just a little dent, and they're all happily making a dent, and the membrane is nicely shaped according to the protein just under the dent, but away from the dent it has the wrong curvature. So there's this kind of bad greenish halo of wrong curvature around this dent, and there's another bad greenish halo of wrong curvature around this dent. Well, what if these guys come together, or a couple of them come together to make something like this? then we have lots of proteins all with the right curvature, and again, just one region of the wrong curvature. So evidently, this reduces the energy, and this should happen. So the very fact that these things are sitting there and curving the membrane should drive them to create bubbles, which is what I want. Turns out this argument is very intuitive and nice, and everyone thinks it's plausible. It's not quite as plausible because once you start to look a little bit more deep into the whole story in terms of differential geometry, the first thing you realize is that the energy of this thing is zero. The green stuff doesn't have any energy, so that argument can't work. In fact, if you go further, there's really no intuitive, good physical justification for why that should work at all. So what you should do is you should say, okay, serious suggestion, let's work it out. Let's do a calculation, see whether this can happen. So here's a calculation. It's from even older, just 12, 13 years old. Michael Kozlov and Helfrich have worked out if I have things sitting on my membrane and then pinch the membrane by a certain angle alpha, and they have a size R0, and they're bending, the membrane has a bending rigidity of kappa, and there's maybe even a tension sigma involved, which gives rise to a length scale lambda. Then this is my interaction potential between them, due to these bumps and halos overlapping. And it's funny, it has Bezel functions and so on, that's yucky. But if you go with a tension to 0, therefore with that length to infinity, all of this simplifies to an even older paper. This is your interaction potential. Really nice, it's a power law. U is blah 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 divided by R to the 4. Problem being that the sign of that power law is a plus, so that's an interaction potential that decays like this. It's a repulsion. Well, that's inconvenient because we kind of wanted to explain why things come together and not why things are being driven apart. So, problem. Another part of the problem is that people have done, around the same time, have done experiments with vesicles, throwing colloids on them. The idea was that the colloids all make little dents, and then the same thing might happen. The dents might overlap, and they might come together, and they do. You have this vesicle, and then all the colloids go to a big colloid raft on one side of the vesicle. And then they roll it. And they also, it's not just the colloids attracting, because if you take the vesicle away, the colloids are just randomly diffusing in your solution. In the presence of a vesicle, they all bunch up on one side of the vesicle. So there's this paper by, blanking on the name. It'll come to me later. This base of PRL, they said, we found curvature-mediated interactions, and they're repulsive. They said we calculated curvature-mediated interaction, they're attractive, and they said they're repulsive. They can't even agree on the sign of these interactions. That's awkward. Was this the signal? Awesome. And then I'm not going to get to the complicated stuff. So we said, okay, this is two parties and they disagree, let's throw in a third opinion, and let's throw in a third opinion using a third technique. One was experiment, one was theory, let's now throw in simulation. And we have systems which are big enough so we can do that actually. So we created one of these toy lipid membranes from which we know the self-assembling fluid, two-dimensional curvature elastic and all of that. We made a really big one, about 160 by 160 nanometers, and we threw toy versions of proteins on them, which are little contact lenses that absorb and make a little dimple, except we can use lots of them because we have that much space. We can control the lateral tension of these things very nicely, so no tension at all, and we can also control that there's absolutely no direct interaction going on between these things, because that's one of the things where experimentalists have difficulties. Maybe there's still some remaining residual attraction between these colloids, and I don't really know, blah, blah, blah. We know there's not because we didn't turn it on in our computer. So that's what we did. So this is our membrane. Here are our little, it's not very much in focus. I'm sorry, I can't do movies very well. So these are our little contact lens dimple thingies. This is my poor man's version of trying to make that bit of the membrane semi-transparent. So let's make it go. So lipids, the membrane fluctuates and wobbles and all of these things diffusing the membrane back and forth, and then you look and see what happens. You see, look at these two, for instance, they stay together very long, that's awesome. I'm going to write a paper. They are, then they fall apart, say, damn, I'm not going to write a paper. But then you realize as this is going on, something much more interesting is going on here. You get this huge bag that's being formed by all these guys being in here. Say, wow, that's what's going on here? But what's going on here is that you have sort of collectively, all of them together are capable of creating all of these membrane bubbles. It's not quite clear whether you have attractive pair forces. We try to see that. It's very, very hard to see. Pair forces, I'm almost willing to rule out. I'm not quite sure, but I don't think so. One thing you see is that this thing that they're sitting all in here, that doesn't have anything like a crystalline structure, which contrasts this to various kind of scaffolding ideas that you have in biology that go with buzzwords such as clustering cages and so on, which have really networks of things being built, and within that network the membrane is scaffolded, and that's not going on here. What you really have is that a whole bunch of proteins, every one of them sharing a little bit of curvature, overall conspire to create this big bunch that in this case, if you map the length scale, okay, you know this is about 4 to 5 nanometers, has about 50 to 100 nanometers length scales, which is correct. This is how big these bubbles are. And the time scale, which is very iffy to map in coarse-grained simulations, our best shot at how long that takes is something like a millisecond or so, which I can't really tell whether this is what's experimentally going on, because these things, if they're 50 nanometer, they're sub-microscopic, and to resolve something sub-microscopic on a millisecond time scale is really, really hard, and I don't think anyone has done it yet. So if you ask people, oh yeah, how long does that take? Say, anything we do in a millisecond in a second. Awesome. So okay, we're not off, but work needs to be done there. Again, this is relevant because what looks like these toy little thingy here, we know they exist in nature. There are proteins which bend the membranes. Okay, admittedly, they came from the other side, they pinch from the outside, my guys pinch from the inside. I don't really think that matters, but okay, maybe we do it at some time, we do it the other way around. There's a bunch of simulation of other people. These people have done this stuff in an entirely different coarse-grained model, put it on membranes, and again, they tubulate and make little bubbles. Again, the third group has taken these things on the membrane, again, different level of resolution, again, curves the membrane, so again, we're doing the right thing. I have we were still nervous about the theory, because theory now is the odd man out. So we did some numerical theory of the whole thing, which I can summarize probably with this transparency before I stop, which said that this is the distance between proteins, this is the force between them. They should all be positive forces, repulsive, more and more repulsive as you get closer. But if these things really bend the membrane strongly, the forces can become attractive. So we have numerically shown in the strongly curved regime where the old theory was wrong, that you can get attractions. But still, there's lots of questions remaining here. I don't understand the whole issue, and that's just bent proteins on a single membrane. It's not clear how that works out. There's a little bit left in my talk on the more highly resolved model, which I will skip, because I think time is out, but I cannot, of course, stop without showing you the people who actually did the work. The people who did the work, the people who did the work are these people. People both at the Max Planck in Mainz, where I started this work, and people right now at Carnegie Mellon, where I am. Those are the people who really do all the work, and do all the suffering, and just they bear with me. There's lots of people who explain to me what I'm looking at and so on. People who give me money, believe it or not. And people like you who listen to all that stuff when I give a talk. And I think that was it. Thank you very much.
We have some questions? Okay?
Yeah, one thing I never understand about the critical points being, the lipid component, the lipid system being close to a critical point is that the only thing that can have RAPs are organisms that can control their own temperature. Is there another?
That's a very good point. First of all, you're not exactly at the critical point. You are maybe 10 degrees off or so. That means you have sort of a distance. And now if the temperature changes, you could of course slightly adapt your lipid composition. And it's known that some membranes change the lipid composition as temperature changes. But the point well taken, if you are an organism that undergoes massive temperature changes, you'd be in trouble if that goes on. Yeah, absolutely. So I'm not saying that it always is like that. There's evidence, there's experimental evidence that if you take plasma membrane-derived blebs and you pull them down, they go through a critical transition. So you could either say that's an entire accident, or you could say there's something behind it. And I'm not the judge of that. I'm just saying if that's true, I can explain a lot of stuff without extra fancy things.
few slides to the simulation of what they call the nature paper.
Which one?
Yeah, right there.
This one or this one?
No, that one, the first one.
Let's make it this one. That's the first one.
Yeah.
Already.
So, if that's a cell, if that were a cell, wouldn't the concentration of proteins be too small? I mean, it's...
Oh, you mean if that were a cell?
Yeah.
Well, that's just, I mean, the cell would be...
No, I get that. But I mean, the concentration of proteins in the membrane. What I'm thinking of, okay, this is a simulation and it's mainly lipids.
Oh, I mean, in some sense, I'm only showing the proteins which right now matter for me. One frequent, possibly gross simplification that almost anybody does who simulates biomembranes is that they either only look at the lipids and not at all of the proteins, or just the one protein they care about. I'm looking only at the proteins I care about. I'm looking at one, and one of them I'm looking at 30 of them, but admittedly there's no other protein in here that could possibly be doing something. And I have not very good justification for doing this other than, well, maybe the other ones don't participate in the bending thing, so they just hang out there, they just go along with the flow or with the ride. To understand what this thing would do if I have like 50 other types of proteins in there is beyond anything I can do. I can just say, if these things do what they do and the others don't do very much. I'm not sure whether this is convincing, but that's the best I can do.
Okay, any more questions? Okay.
Thank you.